Okay, I hope most of you don't mind that I'm sitting down. It has been a long few days, and I think I pushed my leg a little bit too far. So I'm just, if you don't mind particularly, if you can see me, then we're fine. Okay. I always dread being last in anything because I figure there's a portion of people who are asleep. There are a portion of people hoping that neither I nor that the people next to them are, are noticing that they're checking in on their flight. And then there are the others who are just sitting there. They've tried to listen. They think they understand. But I applaud you all for coming, and I do want to thank the conveners of this meeting. It has been, maybe I shouldn't say this, it's been a joy to come here over the past few years. And in this year, even though I'm sitting down, it's a joy to be able to come here. I didn't think that was going to be possible for a while. So what are we going to do? We're going to kind of lessen things down a bit. When I asked Ruben what did he want me to say, he said, I don't know, something about lab tests. And since there are over 20,000 of them, and I know that every now and then people go overtime, but I didn't think that many would go overtime. So let's see. Well, maybe we'll try this for a little while and see what happens. I decided to take the lab tests that are involved with the liver and the kidneys, mostly because everybody has those tests done, and they have them done by many different physicians, and you sometimes wonder why you can't just take the answers from last week. And kind of go through them. I also have to apologize that I can't type. So you will find occasional typos in here, and I apologize for those in advance. Okay, so this is supposed to be that even though we talk about NPNs in terms of bone marrows, and that's in peripheral blood, and that's about it, nobody else really thinks about it, there are other organs that are involved. And two of the ones we're going to talk about. The liver, in addition to doing all of the normal things that it's supposed to do, also is responsible for detoxifying many of your medications and getting rid of debris and leftover material from inflammation, which we've talked a lot about these last couple of days. And so that's an organ that you want to make sure is doing well through your process. And the kidneys are organs that excretes what it can of the waste materials and tries to keep your long-term acid-base balance in order. And while we won't talk very much about acid-base today, it is a long-term issue for the kidneys. One big issue that we will keep coming back to is the fact that the liver, bless its little heart, is the most forgiving of all of your organs. So you can beat it, you can kick it, you can drown it in alcohol, you can do all kinds of things to it, and while eventually it will get annoyed, it keeps coming back to forgive you. The kidneys are, much less forgiving for you. Even less forgiving are your lungs, and as I said, we're not really going to talk about acid base very much, but the lungs are a site of where some of the major inflammatory cells can be located, where you end up fighting off all of your respiratory viruses, and so it's an important thing to keep in track of. And then you've got the stomach that if you've got inflammation will have erosion and ulcers and all those kinds of things. And because we have to include it, even though it doesn't want to be talked about, the brain doesn't like any of this, and is most annoyed at you doing anything about it. Because it is, after all, in control, whether you like it or not. So, what are we going to do? We're going to talk a little bit about the liver and what are some of its functions. Well, one of its functions is to work in digestion. Those of you who periodically have pain in your shoulder or sometimes pain in the center of your, and just below your stomach after a meal of everything you said not to eat. Although I will bet you Jean-Jacques occasionally has a little bit of Strasbourg pate, perhaps. Yeah, that stuff. You need bile, which is produced by your liver, secreted into your gallbladder to be held there for a while so that it can come out and begin digestion of complex fats. We would like you to keep doing that. We would like your digestive process in that sense working well. Ladies, those of you who may have been on or still are on some version of hormone replacement or birth control pills, turns out those particular meds don't do well with the production of bile. And it is possible that long-term use of either one of those could have a problem. So yes, liver function studies are going to be done by your OB. Like, that's not anywhere connected to anything they're used to looking at. The liver purifies things, and I'm using that in a very vague fashion. It detoxifies toxins. It detoxifies drugs. It breaks down harmful substances that you might not have or might not have been good at keeping control of. It will deal with the breakdown of normal waste products to make them capable of being excreted, either through the GI tract or through the kidneys. So you would like that to continue. It regulates your blood glucose. Even as you're sitting there right now, there are some cells. I've decided that they don't pay any attention to anybody else, but there are some cells sitting there in your liver deciding whether or not you need a little bit of boost of glucose now, or maybe we should save the glucose from that second muffin you had before you came in here. And maybe they should store it to a semi-long-term substance called glucagon, or maybe they shouldn't, and how should that be regulated? While at the same time, there is an encyclopedic list of proteins that are being made in your liver at the same time. All hormone regulation is, to a certain extent, regulated or at least influenced by your liver. So those of you who have become tolerant of heat here became that way because your thyroid function decided it didn't need to work that well, and some of that regulation was done in your liver. I, on the other hand, stood there last night in something like this going, why are you in jackets? Because, see, in Massachusetts, it's not warm. So my thyroid thinks this is fine. There are compounds in blood clotting that are also made in the liver, or they are activated in the liver. Many proteins are made as inactive compounds, and when they get to the liver, enzyme reactions occur to make things more active. That is something that if you bring him back next time, I will bet you that Stefan will go into great detail of how the activation of all of these blood clotting enzymes turn into a cascade that create and limit and hopefully then resolve blood clotting, and how that can get messed up if you have any kind of inflammatory process. And yes, there's part of an immune system response that is also positive in the liver, and I think I stopped with this last one. There are more. When you, we'll have a moment of silence here, at 120 days or thereabouts, your red cells move into the spleen, get caught, and die, just a little bits at a time. Hemoglobin inside a red cell, cool. Hemoglobin outside of a red cell, not a good thing at all. It has to be moved over to the liver so that it can be manipulated, so that it can be detoxified and then excreted. And that's just some of it that happen. This is, what am I doing? This is the same. Oh, I maybe got colorful on the second one of these. Okay, so you have liver function studies. They're all the same studies, but Your OB is looking at them from the sense of how close are you to menopause? Are you using birth control pills? Is there a problem with clotting? Because sometimes that's a side effect of birth control pills. Your cardiologist is looking at them from the vantage point of, do you have cholesterol? They don't like any. Poor sports that they are. What is your cholesterol? What type of lipoproteins do you have? Should we control them? How well are they controlled? And is there a problem with stress on your liver because the typical statins that you use are stressful to your liver? And there will be a balance question. How much stress versus how much can we get away with lessening your cholesterol? Your rheumatologist is going to look at liver function studies, again for stress, but also to see things about inflammation. So there are tons of different ways of looking at the same tests, and everyone's going to look at them differently. And sadly, many of my colleagues over here use a word, fine. You, patients, look at this and go, oh my God, that test went up 2 points. And you go to your physician and he says, no, these are fine. Fine for patients when you say it means I don't want to talk about it. Fine when they say it means could have been worse, but I think I'm happy with what we've got. Am I correct? Yes, okay. So, see, we have a communication problem when you two talk, because fine doesn't mean the same thing to the same person. Okay, moving along. So, this is an arbitrary list put in a kind of arbitrary form because laboratories and, sadly, the computer software that some administrator decided to buy make up their own lists. So I didn't want to choose a list and say, this is the way they're always listed, and the top one is the most important, or the bottom one is the least important. So they're kind of clumped together. Maybe enzymes are all together, but they're not listed in a typical patient portal report. So here are some of them. Aspartate transaminase. Will there be a quiz on these names? If this had been the first lecture, maybe. The last lecture, no, I wouldn't do that. What I want you to see is that on this page, these are all enzymes, they're all found in the liver, but they're also found in other places. So you can't say, with one exception, 2 exceptions, oh my God, it's increased, I've got to have a liver problem. AST is found in the liver. Yeah, it's also found in the kidneys and in the heart and in the muscles. So if your cardiologist is looking at this, he or she is looking at this from the vantage point of, are the statins causing this to increase, or did you have a heart attack a couple days ago? Because this enzyme comes out into the bloodstream after a significant amount of damage. So it isn't just a slight little damage off the cell membrane. This is a little bit slower release because you really have to have messed up these cells. So you're not having a heart attack now. If your AST is increased, you had it before. Not hugely before, but before. In contrast, alanine transaminase, ALT, is found only in the liver. So if that's increased, then I know that whatever damage is going on to the liver cells themselves. I don't know what kind of cellular damage, but I know that there's some stress. So you can look at an ALT and say, oh, my liver enzymes are increased. Yes, it is. And it's because of the statin. Well, maybe not. Could have been because that cocktail party we had last night was very nice. Could be a whole variety of other things, but it is a trigger that says, could somebody please look at the liver because something's going on. Now, if the ALT and the AST are together, well, maybe I got myself a liver, possibly muscle or heart issue going on. Alkaline phosphatase, Again, in the liver, it's also in the bile ducts, in the bone and the kidneys. Well, so what? Well, if it's increased and the ALT is increased, then maybe it's more of a bile ducty thing than it is a muscle thing. And if it's the only thing that's increased, maybe we should start looking at the 82-year-old lady who just lost 6 inches worth of her height last year. Maybe something's going on with the bones. So we can see what's going on by picking and choosing the pattern that we see on these enzymatic tests. The worst and the best. The single most sensitive test is the gamma glutamyl transferase, or GGT. Everybody in this room, I betcha, right now has got an increased GGT because you went to the cocktail party last night. And yeah, GGT is increased when you have any alcohol. So the next time you go to your drawing station or laboratory to have liver function studies done on behalf of this side of the room, may I ask that you not do it the day after you go to somebody's house for a big celebration and have some alcohol, because it's going to be increased, and your physicians are going to say, well, how did that happen? It's not supposed to. I wonder what's wrong. And of course, they're not going to ask you, did you drink last night? Because you're going to say fine. And, there's going to be no communication there. And a kind of a positive control of everything is lactate dehydrogenase. It's found in almost every single cell in the body. And if it's within range, the chances are that, there's nothing really huge going on. Okay, we talked earlier today about total protein for a little while. So yes, you can use it to reflect the liver's ability to make proteins. The problem is we use whole blood, serum cells, when we collect your blood specimens. And when we do that, we spin it down, and all of the liquid, the serum in there, is what we test. So what's your hydration level when you have your blood drawn? Because if you decided that this was the day you were going to run a marathon and stop in to have your blood drawn and then continue your marathon, you're probably a little bit on the dehydrated side. And so all of your values might be a little bit increased because you're dehydrated. Or I know some people who have very small veins, and it's very difficult for them to get phlebotomy. So they dutifully pump up those veins. How do they do that? They drink a liter of water. And then they come in and say, see, you can find my veins wonderfully. Well, yeah, I can find them. I'm just not sure I really don't want to take anything from them, because that's overhydration. And now we've got a different issue that things look more on the decreased side. When we talk about diets, you need amino acids to make proteins. Teenage girls, sometimes older women, have eating disorders. They're not going to eat. Or if they're going to eat, they're going to eat 2 blueberries, and maybe a piece of lettuce and say, see, I had a salad. And if that's going to happen, if you have that kind of a poor diet, then there's the possibility you're not making as many proteins as you should, and that will be a surprise to your physicians, because you've told him that you haven't lost any weight and you're fine at 32 pounds. One of the major proteins that you make is albumin. And yes, if there is a decrease in albumin, one of the questions that has to come up is, so how's your diet? Fine. No, I mean, seriously, how's your diet? Because particularly in people, no one here is that old, but in the elderly, particularly those of you who live alone, you can't buy any food in the grocery store because that's for 19 people, and you don't eat that much, right? So you don't buy a lot of meats or a lot of stuff like that because it's just too difficult. And then if you live alone, nobody wants to cook for themselves. They want to cook for somebody else. So what do you have? Toast, tea, cookies. This is a wonderfully well-balanced diet. So the elderly in particular should, if you want to, check your albumin to make sure you're eating correctly. It's not the sole answer. It's not even one of the more important in that sense answers, but it's something you can deal with. You can check your own diet in that sense. Globin is another kind of protein. Actually, it's a whole population of proteins that includes your antibodies. So if your globulin is increased, the next steps, which are not part of the liver function, is to find out which of your globins of your globulins are increased, and that requires additional testing that none of us want me to talk about. So, come on. There we go. Probably the ones you're most concerned about sometimes is bilirubin. Bilirubin is the waste product of red cell destruction. As I said, hemoglobin all by itself is not a good thing to have floating around in your system. But when it is degraded into bilirubin, it can be excreted. In one sense, I'm very glad that John is not here because as everyone on this side of the aisle will admit that there are some pretty sick jokes late in the evening where someone gets paged for bilirubin. Please come to the emergency room, the OB ward, somewhere. He's a very popular guy, Mr. Rubin, and usually when it's been a hard day. Bilirubin is formed into two different components. One is completely detoxified and one is partially so. So there's a direct bilirubin and an indirect bilirubin. There's a couple of things I want to talk about here. Obviously, if you give your liver a huge shock of red cells through hemolysis, your bilirubin's going to go up because it's trying to degrade that hemoglobin. Okay, that's a good thing. Turns out there's a genetic disease where you have a little bit of a problem moving from red cell to indirect bilirubin to direct bilirubin. It's called Gilbert's disease, and it's fairly common and is usually a terror to this side of the room because all of a sudden they see that this is increased and they have no idea why until they figure this out. Sounds like something everybody can cope with. But again, supposing you decided tomorrow that because of the lectures about living a healthy lifestyle, you're going to go home and you're going to run 2 miles, right? Because that's what you're going to do. And the next day, you go and have your blood test done. Well, that turns out to be strenuous exercise. And I know it sounds silly. There's actually a name for one of them that usually happens during basic training called March hemolysis, where the pounding of your feet on the ground, which, guys, occurs in basic training and in other places, the physical pounding of your feet causes some of your red cells to die off. So over strenuous exercise can sometimes increase your indirect bilirubin. Yeah, like who knew? And if you've got direct bilirubin increase, it's probably something more in the liver to blame than it is in the red cells. And that's, again, I'm going to call it liver stress because there are so many things that can stress the liver. I can't list them all. But drugs and alcohol and exercise and different kinds of foods and all of that other stuff goes in there. So what do I want to tell you? I want to tell you that one test alone means nothing. Well, nothing. It could mean something, but I doubt it. It's the pattern of the tests that's important. What did these results say in the light of your OB, your last prostate exam, your last malabsorption test or colonoscopy. What does it say in terms of your NPN? None of these can be done without knowing what your background, your history, your genetics is like. And from a laboratory standpoint, I obviously want to give the physician the single best answers that I can, and I have to depend on you to make sure that you don't come in 2 days after having celebrated, who was it? That you're not from Philadelphia and you're not still celebrating the Super Bowl. Had to think about that. From Massachusetts, we don't think of Philadelphia ever winning the Super Bowl. So, we don't want you, I know this sounds, I'm sorry, but I don't want you to start exercising the day before you come in to get your blood test done. I want you to come in regularly hydrated. I want you to come in at about the same time of the day, because different ones of these tests, of these compounds, will vary during the day. So, if you were diagnosed with a blood specimen at 8 o'clock in the morning, could you give them a break and always have your blood test done in the morning so there's a consistent level to them? Because then that way you give me the best specimen, I can give them the best result, and they can figure out to a much more consistent level what's going on. You'll notice on this, I said, an increased GDT with absolutely no other finding is a glass of wine 2 days ago, but it's also an aspirin. Now, some of you are on everyday aspirin. Different story. Everyday aspirin means that it's consistent from day to day to day. But if all of a sudden you're not on aspirin, you get a headache and you take 2 aspirin, please don't get a GGT done maybe for 3 or 4 days. Because there will be a skew to that result that will confuse them. And the last thing we want is to confuse them. Right? I mean, that's a good answer. Okay, what about the kidney? Much fewer functions in the kidney. But, as I said earlier, the liver can repair itself. I mean, think back to the earlier days. Was there someone in your college dorm that maybe smoked something? Not you, of course, but you were walking by that room. Or someone else was doing something, and I don't know, your next door neighbor had a challenge to see how many 6-packs you could drink in a half an hour. You really need a trained liver for that, by the way. Those kinds of things your liver will repair. Your kidney does not repair as easily. You got 2, you only need part of 1, but you really want to keep both of them functioning for as long as you can. So what does it do? It filters the blood of waste products. You want that filtration to happen at a very efficient level so that it's constantly moving. There's no push on it one moment to another. You want it to maintain itself for consistency. It balances your sodium and potassium levels as part of its fluid function. It maintains phosphorus and calcium for your bone health level. And it participates in pH. So it's got a lot of work to do, just not as much as the liver. So let's see what happens. Albumin is tested in a urinalysis and in blood tests because albumin is not supposed to be lost through the kidney. So if your kidneys are losing albumin out into the urine, then there's something wrong with a part of the kidney that's the filtration unit. It's called the glomerulus. So you not only have a sense then with the albumin of that the kidney might be in trouble, but also the location in the kidney that might be under trouble. The blood urea nitrogen is a catch-all test. Who has nitrogen in their compounds? Well, a lot of proteins do. And so you want the ability of the liver to break things down in sufficient quantity so that the kidneys can excrete some of that urea nitrogen. That is probably elevated in everybody who went to an absolutely phenomenal dinner last night at Lawns, because there was fish, that's got a protein in it. There was the filet mignon. There wasn't pate, but we could fake it. There was a lot of high proteins. You remember the old movies, Who Got Gout? Rich people got gout because they ate a rich diet. Your blood urea nitrogen can increase if you're eating a rich diet. Creatinine, on the other hand, is a waste product of muscle metabolism. So, women under 5 feet probably have a slightly lower creatinine than one of the guys that's 6 feet tall. Women who don't exercise will have a slightly less creatinine than somebody, male or female, who spends an hour in the gym every day. So it's a muscle metabolism. It's a pretty constant kind of number. But if the kidneys can't function that way, then you're going to have a buildup of that. There we go. You're going to have a buildup of creatinine in the bloodstream, and you're not supposed to have that. The kidneys are supposed to express that. Renal damage, particularly the other part of the kidneys called the tubules, have problems with that. So do any medications that none of us take. Anti-inflammatories, antibiotics, beta blockers. Nobody here takes beta blockers. Anyone who was pregnant probably had an increase, an augmented creatinine level. Reduced muscle mass is lower. The vegan diet will give you a lower creatinine. So when you go on her vegan diet the way she wants you to, and it's 2 months later, and you say, oh my God, my creatinine is down in half. Good. It's a response to the vegan diet. So it's not such a bad thing. The EGFR, people in medicine don't like to let anybody else know what's going on. So they invent names like estimated glomerular filtration, which A, doesn't mean much when you think about it. And then just in case you actually looked it up, we use the abbreviations. EGFR. This is trying to tell the physician approximately how good that glomerulus is in filtering what's coming through the kidneys. The real GFR requires you all to get a 24-hour urine specimen. All those in favor of that? So why don't we do an estimated one? We'll take a random sample and add in some mushy factors and make a calculation and come up with a number. The number is typically printed over 60. That does not mean that if you're 100, you're 40 times better than the person who's 60. It means you got a really good filtration rate and you don't have to worry about anything messing up with the kidneys immediately. But what about those of you who have got a 52, 43? You look it up on Google and it tells you you have terminal kidney failure. How about you're over 70? When you age, the glomerular filtration rate will drop with, age. When else can it happen? Medications that will play around with or try to influence the degree to which fluid is moved around in the body. What do I mean? Diuretics? Nobody here's on diuretics, right? I mean, that's not one of the things your cardiologist ever puts you on. So here you've got a situation where the EGFR is influenced by age and two of the most typical classes of medications used in cardiology. If you don't tell them what other meds you're on or what other conditions you've got, then they can't interpret that EGFR correctly, because they're going to look at a 43 and go, oh my God, she has kidney failure or is heading towards it. No, you're not. I mean, we messed around with it so it's low, but this is another plea. Let them know everything you're taking, because even though they're not involved with NPNs, they are involved with you as a patient, and that's important for you to do. Now, there are some people who like simple answers, and so one of the things that has been invented is a series of ratios. How much albumin is there in your urine compared to how much creatinine? How much in your blood, how much blood urea nitrogen do you have in comparison to your creatinine? These are ratios. They're, what's ratios? 5th grade math? Maybe? So you're gonna probably have to think about it. But just remember that because there are so many influences on the BUN and so many influences that can come in on the creatinine, a ratio has to be taken with a gram of salt. I wasn't really planning on that, but I think I'll move on fast on that one. Sorry. So, I'm all of a 5 minutes. For the first time, I think I'm late. So what am I telling you? You all have to make sure that you are in the most consistent state that you absolutely can be before you come to me. I'll handle the testing. They all need to know everything you've tried in the last 48 to 72 hours so that they can properly interpret both the liver and the kidney failure results. Because otherwise they're going to think you're in some kind of failure or distress when really you're taking drugs as directed. So you both need to talk to each other, preferably in, well, I was going to say English, but English or French or German, in such a way that you can communicate what you're doing on the outside so that they can determine more fairly and more completely what's going on inside. Okay, there were a few people nodding off, but most of you were okay. I'll quit while I'm ahead. Thank you very much for paying attention.